Hi, this is Ramji Srinivasan, co-founder and CEO of Teiko, and we're going to talk today about a question that a lot of customers who are new to high-dimensional cytometry ask. They say, don't you just do cookie cutter gating? And more specifically, they say with up to 245 populations on these 25 marker spectral flow panels, or 41 marker panels, how can you possibly gate all of these populations? You must do cookie cutter gating. And the short answer is no. We enjoy our beating, so we individually gate even for 25 and 41 marker panels. And you can get a sense of the scale of this problem. On the x-axis, you have different panel sizes from 13 markers to 25 markers to 42 markers. On the y-axis, you have the number of immune population analyzed. In a conventional flow with 13 markers, that's about 100 populations. Spectral flow is 245 theoretical populations, and as you get to mass cytometry at the upper ends, you can get to 700 plus populations. So that's got to take a lot of time to measure all those populations, and it does. So let's just take a 25 marker spectral panel as an example. That has 245 theoretical populations, of which we analyze 146 in our CLIA validation. And there are 82 functional markers where we would look at on a case-by-case basis, and 64 gates per specimen. These gates, we look at all 84 specimens, and that yields 5,376 plots, which takes about 10 hours to analyze. So 10 hours seems like a lot, but then a careful reader might notice at the end of this that that's only about 7 seconds per plot. How could we possibly get it right? Well, the short answer is heuristics. They're a real time saver. So if you look over here on the x-axis, you have the number of samples, 1, 25, 50, 84. And the y-axis is the time per gate in seconds. So you can see in the beginning, if you just do one sample, it'll take a lot of seconds to do a gate. But as you get more and more samples, this time per sample goes down. But how? How does that work? Well, let's take a sample gate. This is CD16 and NK cells. And for a single patient, you just draw a boundary over a single gate, and you can see that gate shows up correctly. And if you were to do this for 2 patients, so again, you do NK and CD16, and you draw this same gate, it appears across a second patient. So already you're seeing a time savings, and you can visually inspect this to make sure it's in the right place. And then as you get a third gate placement over here, or if you have more patients, you draw that gate and it immediately propagates all across from patient 1 through patient 6. And now it's easier by eye to go from left to right and inspect all these gates and make sure that they are within the right setting. And we don't stop there. Every single gate placement is independently reviewed by another scientist. So you can imagine as you go from patient 6 to patient 40, 70, etc., it starts to get a lot faster even though there are a lot of gates. Now, let's take what happens in the cookie cutter method. In the cookie cutter method, you just draw that first patient gate and you immediately copy and paste over. And even though it's in the wrong place, as you can tell over here, even if you don't know anything about flow cytometry, this gate is right in the middle of a contour plot. So this same gate is applied here, whereas with Teiko, you have this gate over here, and then through inspection, our scientists know to adjust this, and this gate is what's known as tailored, and you get full visibility into this tailoring, as we'll talk about. What's a consequence for clinical development? You get the wrong number of cells with the cookie cutter method. So patient 1, you had 85%, that's correct over here with Teiko as well. Patient 2, this was 76% cytolytic NK cells out of total NK, and it's a 12% increase on the right-hand side. Using cookie cutter gating would have missed 12% of these cytolytic NK cells due to these varying levels of CD16 between patients. So this is why cookie cutter gating is bad and can potentially lead to problems. So how does cookie cutter gating work? And we put this in quotes because it works badly. You start with the panel, probably off the shelf. You apply this cookie cutter gating strategy from old school CROs. You get this strategy with no tailoring, and then you get a bunch of messy, ugly data. And this is you with the unhappy face after you see all this, because then you have to go back to the CRO and potentially argue with them to try and move gates, and they can't do it, and it's inflexible. And as a result, you just spent a bunch of money on a flow cytometry project that needs significant rework. How do we do it at Teiko? You take a panel and you get either custom or standard. For purposes of simplicity, we'll just focus on a standard panel. Standard panel has a gating strategy, and even in that standard panel, you're able to review that and you can inspect and make sure that all the gates are where you want them to be. You could potentially add gates like CD8 gate to NK cells. We'll incorporate that. Then we'll do the next step called post-processing, and this is where we'll do all this gating and tailoring. And this is a crucial step because if this isn't done, you'll get a gate that just gets copied and pasted, and you'll get a lot of bad junk data later. Then we'll have another scientist do an independent review of that first scientist's work. Ultimately, you'll get gated data here. You can look at this, inspect and make sure that all the gates are in the right place, and then you're able to give your approval. And then we proceed to the data analysis step, run everything through our engineering pipeline, put it on our dashboards, make it super easy to use. And you can see that frown just got turned upside down. So this is what we do at Teiko to make sure that you get the best data for your cytometry projects. The benefits along the way are complete visibility. You have flexibility in terms of the option to add or remove gates during panel development, and transparency/visibility. You get a peek in at your fully gated samples, allowing you to make adjustments to tailoring if you need to. So why do customers care about this? It's basically no surprises. It shocks us to hear how many times scientists are wasting time trying to redo results from their flow cytometry vendor. And as a result, they are not making progress on their drug at the pace that they need to. So the other benefit for customers is no badly gated data. You get good quality stuff that works right out of the box that you can present to other scientists, to investors, regulators, etc. So if this topic interests you, subscribe to our YouTube channel for more cytometry insights, or visit teiko.bio to learn more. Thanks for signing on.